Hello, my name is Liliana Mandujano, and today I'll be presenting on how proboscis of the Apis mellifera is used for pheromone exchange during trophallaxis. So first, let's briefly introduce the species. Apis mellifera, or the honeybee, most known as Western honeybee, is a eusocial species, meaning individuals in the colony share labor and cooperate across generations. The colony includes three main castes, which is the queen, workers, and the drones. One key feature of honeybees is that the workers progress through different roles as they age, from cleaning and brood care to guarding and eventually foraging. Their entire colony functions through chemical communication, especially pheromones, which regulate everything from task division to reproduction. Next, we will be going over the background of colony communication in the honeybee colony. So, chemical signs or pheromones are essential because they help regulate colony organization. Some pheromones work instantly to trigger behavior, while others shape long-term roles and physiological responses. Some pheromones spread through air, but many, especially the ones tied to social structure, require physical transfer between bees. This is where trophallaxis becomes important because it ensures the colony stays coordinated and stable. To understand how pheromone transfer works, we need to also understand the structure involved, the proboscis. The honeybee proboscis isn't just a straw, it is a complex mouth structure made of the labrum, labium, and the glossa, which together create controlled canal for liquid transfer. The surface also contains the sensilla, which acts as a chemical sensor which allows bees to detect the composition of whatever they're exchanging. This allows them to not only share nutrition but also communicate through tiny droplets passed mouth to mouth. So now let's define trophallaxis. Trophallaxis is a direct exchange of fluid, food between bees, and it requires close physical contact. While many people may assume this behavior is only about feeding larvae or distributing nectar, it is also playing a major role in communication. Through repeated exchange, trophallaxis helps move chemical signals efficiently through the colony. This brings us to the main research question for my project. How does trophallaxis aid in the spread of pheromones in a colony? The first study examined how the queen mandibular pheromone, or QMP, spreads among workers. Workers were exposed to the pheromone and then observed to track how behaviors like grooming and trophallaxis contributed to its movement throughout the colony. Researchers then sampled the bees to measure pheromone presence in different individuals and body regions. Results showed that QMP spread far beyond the original exposure site, demonstrating that its movement depends heavily on social interactions. The more the bees engage in contact-based behaviors, especially trophallaxis, the more widely the pheromone became distributed. This confirmed that physical transfer is a key mechanism for queen pheromone distribution. As you can see in the non-grooming versus the grooming body parts, in the mouth parts versus the thorax, legs, and the abdomen, you can see the difference in the distribution of QMP across the body regions in the grooming versus non-grooming worker bees. The next study I focus on trophallaxis as a social network. Research found that trophallaxis patterns aren't random. Certain bees act as hubs, meaning that they interact more frequently and play a larger role in distributing resources and signals. The study found that worker age and role influence how efficiently the network spreads resources and pheromones. The patterns formed through trophallaxis help regulate both nutrition and communication, showing that these networks provide decentralized control rather than top-down hierarchy. Together, these studies show that trophallaxis is not essential for just feeding, but is essential for colony-wide communication. It allows pheromones, such as the QMP, to spread readily and quickly, supporting colony coordination and task division and overall stability. In summary, trophallaxis serves two purposes: nutrition sharing and pheromone distribution. The structure of proboscis allows precise fluid handling and repeated social contact, ensures that pheromones circulate throughout the colony to maintain organization. Understanding this behavior has practical value, especially for beekeepers. Knowing how pheromones spread can help with the colony monitoring, hive management, and especially the successful queen introduction and interaction. It also expands our understanding of how complex social systems emerge from simple repeated interactions. Thank you. That is all for today, and I will take any questions, and these are my references for my presentation today.